As Dr. Hoffman said, I went to North Carolina A&T, did an undergrad in animal science and then a graduate school in animal health, and then on to NC State for MS in plant pathology, and then here to Tuskegee in 2015, graduated. And I joined as faculty in 2017, so I'm very fortunate to have a job, particularly in this time of in this state of our world and our reality. And so you see the HBCUs, the historically black colleges and universities, have recently taken a lot of, have gotten a lot of exposure. Life itself is based on diversity. Genetic diversity drives longevity. And that's for any living organism. And so for a living organization, diversity is very much necessary in order to continue to promote and to continue to build and also to utilize all of the powers, the talents that different people have from different backgrounds, ultimately to bring those powers together to accomplish a major goal that benefits society. And so I'm appreciative for this opportunity. And so some of the work I did at North Carolina A&T State University with my beloved mentor, Dr. Malumabet Worku, she was really my first mentor and one of the greatest mentors I've ever had. She's incredible. And so we work with meat goats and intestinal parasites that will cause constraints on meat goat production for farmers, particularly small scale farmers, local farmers in the area of North Carolina in Greensboro. The first publication actually was in 2009, and so with Dr. Worku and Dr. Ametna, and so we looked at the diatomaceous earth, which is a product of these diatoms, these single-celled algae that are found in the ocean. Millions and millions of years, there's been a buildup of these organisms on the seafloor, on the ocean's bed, and so um, properly mined, the outer surface of these organisms will form a silica. It's really silica base, which is like glass, the component of glass. And so the when dried, the powdered form of their outer surface, this silica based outer surface, provides a mechanical abrasion. And so many people have used diatomaceous earth. They sprinkled it around their houses or farms or indoor places in order to provide a biopesticide, a natural biopesticide. So as the insects are crawling over the diatomaceous earth, they are being ripped apart by the silica nature of the dust itself. And so we wanted to see if there's been many reports that said that diatomaceous earth could be used to control intestinal parasites. So we actually performed some research to test this as a method of developing a biopesticide, a very safe. And we're in a time now with very green engineering. And so we're looking to be environmentally conscious and make sure that we reduce our impact, particularly in major production, farming production, because there's so many different chemicals that are added to our environment, and that can cause an environmental impact. So how can we mitigate pests and develop pest management techniques that are actually safe for our earth and our environment and for our future citizens? And so, unfortunately, we did find out that this treatment did not work because it's based on dryness. And so, when we drenched the goats with the diatomaceous earth, of course, it became moist, and so that you lose the mechanical abrasion, the entire mechanism of action. And so, but still, to students looking to get into academia, even if the information is not somewhat positive, a negative result is still publishable. And so um, you can also show people what does not work. And so that can be very, very important for disseminating research to stakeholders and people that actually need this knowledge. Because a lot of people were testing this or using this and saying that it worked, but it hadn't been scientifically proven. And so the information we gave hopefully has dispelled some of the myths and beliefs. After graduation from um, North Carolina A&T, I did a year of post-bac research at Virginia Tech, and I worked with Dr. Zhang and Ying Wang and Satyam, the graduate students in the lab. And so we did pronuclear microinjections. So we inserted genetic constructs into the genomes, the nucleus of mouse embryos, and then implanted those embryos, of course, into surrogate mothers through the infundibulum, sold them up, and then they would give birth to transgenic pups, which are the mouse babies. And so then we would analyze to see if our gene has actually been inserted into the genome of these animals. And so the learning the technique of pronuclear microinjection and all the techniques I learned from that experience has been very helpful in my professional development. And so moving from Virginia Tech, was accepted to North Carolina A&T, North Carolina State, and where I work with the fungal genetics department. And so Dr. Mitchell, I believe now, who's at Iowa State, we worked with elucidating some of the molecular causes of pathogenicity in a major fungus, the rice blast fungus, Magnaportho oryzae. This fungus kills enough rice to feed about 60 million people. So that becomes a huge global food security threat, and particularly for countries that are rice is a staple food. Your Asian countries, African countries, this becomes very, very important. So as a disease, it's very, very serious. But as a pathologist, I guess sometimes you get a little excited about looking at the different types of diseases. And this whole rice blast, literally, once the fungus infects the rice field, it looks like a bomb has blown due to the devastation of the crop. And so it's a very, very important pest. And it's important that we understand the whole host pathogen interaction. As the pathogen evolves new proteins or molecular factors to infect the plant can't just get up and run away like we can when they're under attack. And so they must also evolve and have elicit defense responses through the production of proteins and enzymes and other chemicals to actually prevent the infection process from pathogens. So there's a evolutionary arms race that's been going on for millions of years between pathogen and the host plant. And so we, as researchers, our job is to, of course, jump into this war and help the host plant, particularly our crops. But then, of course, we also are forcing the evolution of the pathogen as well. So that's something we always have to consider. And so agriculture is more than just about cow and plow. In the 80s and 90s, Tuskegee partnered with NASA on the Controlled Ecological Life Support missions to actually send sweet potatoes into space. So at Tuskegee, we're mandated to work with sweet potato and peanut due to the legacy of Dr. George Washington Carver. And so we actually sent sweet potatoes up into space as a measure to not only provide oxygen potentially for the astronauts, but also a food source, and also to study the type of lack of gravitropic effects and the different growth and developmental processes of sweet potato under hydroponic growth. And so it's a very exciting project that we're hoping to resume now, as NASA has really kicked up their thrust to explore different planets, their extraplanetary studies. And if you all have seen the movie The Martian with Matt Damon, you know that the first way of colonizing a new planet, of course, is to plant crops, is to start agriculture. And so I work with plant parasitic nematodes primarily now, such as Melododon incognita, the most important plant parasitic nematode in agriculture. Plant parasitic nematodes cause billions of dollars in yield loss each year. And their method of infection, particularly the root knot nematode, is damaging because it infects the root, the primary source where the plant is actually obtaining water and nutrients from the soil. And so these are some of the video I'm taking. And as you see here, the nematode are starting to molt. So they're hatching out of the eggs, as you see here. And once they hatch, they would then, at the J2 or juvenile 2 stage, they will migrate through the soil, and they're attracted to the chemicals released by the plant root. Now, once they enter the root, as you see here at the bottom left, the proteins and molecules that the nematode is inserting into the root cell causes extensive endoreduplication without cytokinesis. That is DNA replication without the partitioning of the new DNA into new daughter cells. And so they're creating a giant cell as a feeding source. And so the symptoms of infection, of course, are the knots, hence the name root knot nematode. And so this causes tremendous economic losses for producers. And so we have to find ways to actually mitigate this type of pest damage. And so I screened some newly developed sweet potato cultivars from Tuskegee and also some pre-existing cultivars to see if they had genetic resistance to nematode burden. And so we were able to select some of the genotypes that have nematode resistance. And so we were actually able to do some preliminary work in analyzing the differential expression of proteins and genes that are involved with the defense response to pathogen infection. And so we see that these NBS, ARC, LLR, these are domains found in plant resistance genes, resistant proteins. And we can see that the levels increased in the resistant variety as opposed to the susceptible variety during the study. And also doing some protein analysis, I was able to identify that there were mutations actually in the amino acid sequence, which may have led to the less expression in the susceptible sweet potato genotype. And so moving forward, recently, I've been working with one of the grad students, Dr. Naresh Shahi, and of course, my mentor here, Dr. Egnan. She was my mentor during my PhD work here, and Dr. Min, Dr. Bonzi, and Dr. Collier. So we developed silver nanoparticles using a very green method using chitosan. Chitosan is derived from chitin, which is found in the exoskeleton of a lot of crustaceans, and also it's the primary cell wall component of fungi. And so we use chitin as a reducing agent to develop these silver nanoparticles and actually use them to expose them to the nematodes. And as you see here, the nematodes on the right were dead within a few minutes. And so the efficacy was incredibly strong for the use of these silver nanoparticles. Hopefully, with further study, we can develop some sort of a biopesticide that can help farmers alleviate some of their pest concerns, particularly with the nematode. So recently, we were awarded a grant to study the effects of magnetic fields and actually in plant growth and development. So I started a research team of students, and it's really cool because the grant, we had enough money. We really targeted our money to provide stipends for students. So they get research, experiential, hands-on research training, and they get paid. What undergrad student doesn't want to get paid? So yeah, I know it makes me feel good to be able to give them stipends each month. And they've done a great job with their research. And so this is the Helmholtz coils that actually we use to generate a magnetic field to apply specific magnetic field frequencies to our plant seeds in order to hopefully boost plant growth and development and reduce the amount of chemical fertilizers that are used in crop production. And so we're working on that now. I'm getting some pretty good results. And so this is our new research unit that was we just installed with the grant money, so students can have a nice, safe, clean environment to do their research. And named it the Carver Phyto Research Unit after George Washington Carver. And so our program that we're starting is called the TU START. That is Tuskegee Student Agricultural Research Team, because you have to have a team in order to achieve victory. So we have a little bit of preliminary data that shows that at specific magnetic field frequencies of 2.5 Gauss and maybe 8 hours, we see that in carrots, or we see an increase in root and shoot length due to our magnetic field treatment. And in collards, we're seeing some interesting trends as well due to the different time intervals and the frequency, the actual magnetic field frequency that we are actually exposing to the plant seeds. Very recently, myself, my incredible department head, Dr. Olga Bolden-Tiller, and Dr. Curie, we received a grant along with in conjunction with the University of Illinois to actually implement artificial intelligence (AI) here in Tuskegee. And so we're hoping to increase our research infrastructure with the incorporation of AI assisted learning. So we build our students to be modern professional agricultural workers that are skilled and knowledgeable of the latest techniques in precision farming, and also the incorporation and implementation of AI assisted learning. And so one of our major objectives is to adopt the AI assisted learning tools and help to increase our research infrastructure, teaching structure, and help to better train our students to be proficient in the modern agricultural research techniques. And as you see here, I'm really interested in adopting this Agrobot. As you see here at the bottom, that can actually go through the field and take real time information, phenotypic information that we can actually use AI to analyze. And so I've named this the Dr. Garish up here at the top. He's one of the people who's innovated and developed these Agrobots. And so I'm actually trying to get him to let me name it the AI rat. So response assessment tool. So I thought that'd be a pretty cool name for this Agrobot as it's moving through the field and taking phenotypic samples, actually measurements, and performing phenotypic analysis. And they've even engineered one that actually has hands that can actually harvest as it moves through the field. So we're very excited about getting this technology, introducing it to our students, and working with our small scale farmers. A lot of the work we're doing now, we're working with small scale farmers because, as Dr. Hoffman talked about, is dissemination is so important. It's not only that we develop and innovate and provide research for producers, stakeholders, and society, but we must get it to them. You know, we have to perform the extension, the outreach, in order to get this new information to the people who need it the most. This is a device we're developing now with Dr. Mehta. This is a automated phenotypic system. And so we will incorporate the AI assisted learning with this. And as you see, these will be fully automated. So we're going to introduce this into the CPU. I call this the AI PRAT, the AI assisted phenotypic response assessment tool. So we'll have cameras that will be moving in real time and taking pictures of things and recording leaf area index and other phenotypic assessment tools that we can actually use when we're performing research and trying to assess treatment effects due to our research. And so you all are the trailblazers, the future, the next generation of intelligent, skillful, male, female scientists. And so we encourage you all to really dig into agriculture and realize how important it is and use your talents to help benefit society. Thank you.